Helen, take it away.
Thank you. All right, so I'm really just going to talk about some old stuff, and I hope I won't go on too long. Most of my talk's actually on the first slide, and I really wanted to, especially for the younger people in the field, I wanted to just give you an idea of what it was like, how CCP-EM has really changed things, because first of all, where did it come from? So the MRC had written all this software, all these Fortran programs for image processing quite early on, and the libraries and everything were the basis of what it became CCP4. There were a few little hitches, like if you try to, once we started using CCP4 and you tried to import an EM map into it, everything crashed because the CCP4 didn't like space group 0. And I was really stuck by that for a while, but Martin Wynne came to the rescue. I think I grumbled to my colleagues at Birkbeck, and Martin was somehow recruited to fix this. So he fixed the library so that we could look at EM maps in CCP4. So that was quite good. Eventually, Steve Fuller and I persuaded Richard Henderson and Tony Crowther to unify the formats a bit more, because things had drifted apart a bit, and it made it quite difficult to switch between the EM and the crystallography software. And we needed both. So that was quite helpful. That was all sort of prehistory, the woolly mammoths had just about stopped roaming Trafalgar Square, etc. And the PDB was a huge example. So both CCP4 really showed the way for how to have a community-wide, there were some very sort of community-minded scientists, particularly Eleanor Dodson, I think, who decided that all this software should be unified. There should be some common way of having an interface and having training for the whole field so that people could share and develop things in common instead of all just doing things in their little corner separately. And that really was the model for CCP-EM. And it was a terrific model, and it's made the CCP-EM development was much, much faster because we already had the template for how you should do these things. And the same with PDB and EMDB. It was clear you needed a public database so that people could deposit their stuff, developers could look at things, people could evaluate the whole field and all that. So EMDB and eventually Empire were really based on the ideas of the PDB. So all that's been really helpful. And then having central facilities like Diamond, synchrotrons, Daresbury and then Diamond, also made it clear that if you had a big expensive resource it did seem a little bananas to do that for EM at the beginning, but it seems to have worked out fine. So having centralized resources is also a very good thing. So CCP-EM, for those who can't imagine, it's hard to imagine what it was like before. Everything was quite difficult. It took all the time and effort just to try and get the methods to work. It was all, it wasn't just the software, of course, it was the EM as well. But you didn't have time to really work on the biology so much. It was, it took a lot of time and effort just to make the methods work. And there weren't too many people nuts enough to do it in those days. So it meant we all knew each other. It was quite nice. But everybody else thought we were bananas. But that was all right. It was fun. So now with CCP-EM, you can really focus on the biology. You can really think, what are the interesting problems, or what are the interesting methods I want to develop? And it's all there, for a user, for a biological user, you can kind of press buttons and things just happen. A lot of things just work. And as you heard, the developers are really trying to make it so that things work more automatically and more easily and do all the technical things we need so that the users can just get on and focus on interesting biological problems or whatever they want to do. So one thing that I think we'll have some discussion tomorrow, and I thought it might be nice to stimulate some of that discussion now, is where should it go? Where should it go next? But maybe we'll come back to that at the end, unless somebody wants to already remark on that now. But okay, yeah. So I thought I'd cover some of that old history, and then maybe just show an example of my work on cryo over the centuries, and see how that changed with the changing methods. So this is, I think you're all familiar with this sort of idea. This is just covering the range of things that the field can cover, starting with fluorescence, obviously, and going all the way to atomic structure. And CCP-EM really has covered most of this. Here. Can you, because my pointer visible, it might be easier if I just do it on screen. So most of this is covered by what CCP-EM does. The microfluorescence imaging is a tricky business. Trying to have a database and a system for really looking after all the fluorescence images is more tricky. That's a kind of separate operation, but we do need to link to that to plug into correlative microscopy. So how did the EM side of this start? And I think it's always fun to go back and look at what was recognized early on. And in particular, you know, it was all very obvious really early that the radiation damage was a big deal. And that was this old tomography picture showing the chicken being roasted as it was being imaged. And then there was the business about projections. So, you know, people realized, well, these are recording projections of 3D structures. And then I think this is, I'm pretty sure from one of Joachim Frank's reviews or books. And the ambiguity of what if you have a single projection, you just don't know. The vertical projection of this structure and this structure are the same. These two particles would look the same in the top view. So this is a really serious problem when you're trying to sort out a whole jumble of orientations and a whole jumble of structures and continuous variations and all that stuff. That's what people are, you know, struggling with, to get all those things, to sort out all those things at once, because they're all jumbled together in our data sets. So that's sort of where we were at the time. This is, you know, 1970s. I mean, all this started in, I suppose, the late 1960s. I became aware of it in the 1970s, 80s. And the thing that made everybody think, well, maybe EM can do something with biological structures, something serious with biological structures, was when Richard Henderson and Nigel Unwin showed that you could take this purple membrane, this bacterial membrane with this 2D crystal of bacteriodopsin in it, and get diffraction from images where you could see little tiny diffraction spots. You may not be able to see them, but they're there. And then filter out all the noise in the background. So the images look blank. You can't see the structure on the image. It's too small. There's too little signal. But you could filter out all the noise between the diffraction spots. And then they got this amazing projection map showing the alpha helices and projection of these trimers of bacteriodopsin. And that was a really stunning demonstration in the 1970s. And that triggered the idea that, well, you know, if you could really get these methods to work better, you could look at, you know, structures of proteins, structures of macromolecules. So that was quite spectacular. And they did tilts and got it to 3D. Now, I should say, doing a tilt series on photographic film is not for the faint-hearted. Most of you, I think, couldn't imagine how ridiculous that was. And you had like 25 or 50 films on a cassette, and you know, you had to. There were all sorts of vacuum pumps involved because the films would be all wet and you couldn't put them in the column of the microscope without making a mess. So this was, you know, pretty inconvenient. But that's what we had. You know, we didn't realize there was anything better at that time. And then the ideas of it took, well, there was a really separate line of development to lead for single particles and finding objects not in a lattice. But Richard had this, and his colleagues had this idea of unbending, where they had a lattice with, and it's exaggerated here, but the 2D crystals weren't ideally organized. They weren't perfect crystals. There were some wobbles in them. And what they did was you could use correlation. You could say, take images, little patches, and do cross correlation to find out where the things really were instead of where they were supposed to be if they were in a perfect lattice. And then you could jiggle it, you could interpolate it to make it into a perfect lattice, and then they'd have nice diffraction that could carry on as if it was all a proper crystal. And so, but those ideas, and I would say Owen Saxon was somebody really early on involved in using cross correlation to find particles. And that also was the basis of this single particle idea, which eventually became much, much more broadly applicable than the 2D crystal one, which is hard going. And then the idea of classification started, well, probably before this, but so Van Heel and Frank, who didn't subsequently work together, but they did then, came to the idea that there was this guy, Breteaudier, that had suggested you could use statistical analysis, multivariate statistical analysis, to classify things into subsets. And so you could show this by, say, this is an image with 2 pixels, a dark pixel and a light pixel, or a darker one and a lighter one. And you could represent that classification in a 2D plot. So that's quite nice. You could do a 2D plot and you can see there's clusters, there's different patterns that come out. Now, if your images nowadays might have 1 million pixels, and so you'd have to have a 1 million dimensional plot. So that's harder to show in slide, but the idea is the same. And so you can show that, you know, you have a multi-dimensional plot that shows you where your different clusters of images might land. And so one of the early applications for it, and I have to say, when I looked at this at the time, I thought, this is really boring. Like who cares about the staining of hemocyanin? And I just thought, oh, you know, but so I didn't realize how useful this was going to be, because, you know, they used it to show they could classify, you know, up and down for different staining variations on this hemocyanin or whatever it is. So, you know, and there were 2 clusters and they could show it. So I was a bit of a barbarian and didn't appreciate it at the time. Anyway, the other thing that was really important was vitrification. So purple membrane is this very unique stuff, this 2D crystal. It's like concrete. You could just take those sheets of membrane and put them in a high vacuum and they wouldn't disintegrate. So that's the only biological object practically I know of where you could just put it in a vacuum and it's fine. So nothing else is like that. Of course, everything else has lots of water. And Jacques Dubochet showed that you could, if you just plunge things into a proper cryogen like liquid ethane, you could get the water to go into a non-crystalline state, just to vitrify and not have ice crystals. And then if you, at higher temperatures, you get these ice crystal forms. And this is a demonstration. This is a vertical section through a pellet of red blood cells. And this is to show, and the top part is where it hit the coolant. I don't know if it was a copper block or something like that. And you could see the red blood cells are fine at the very top, and the further you go down, the more they're damaged by ice crystallization. So this is the demonstration that you can only vitrify a very thin layer. And so that made it very clear. And then if you have a high pressure freezer, you can get deeper, which is why we need high pressure freezing to look at pieces of tissue and thicker stuff. So that then the ingredients were kind of all there at that point. And we're still, you know, in the 1980s, this is still, well, this was the beginning of the idea of cryo-EM, you know, it's just starting then. And then there was this idea you could get single particles and use this cross correlation to align them. Now there was lots of noise. Neil will remember this slide because I took the picture, but he made the rest of this stuff. Even though I think it's copyrighted too, even though Gal is online somewhere. But, so yeah, so you have a field of particles, and it's going to be noisy, it's going to be hard to pick them out, but they are there. And if you have some kind of crummy starting model, but that they vaguely correlate with, you can use the cross correlation to group them into classes that sort of match, and then you could average out each group and you get a better 2D average, and then you can go back to 3D and get a better 3D model, and you can iterate this. So that was pretty good. And that also, I like to use this to explain that that's why there was a quite strict lower limit to size, because if there was a mouse in the field, you wouldn't be able to find it in this noisy image. So you had to have big enough stuff to do this on. And so one of the early things that, I mean, the best samples at that time were icosahedral viruses, because they were big and they had lots of symmetry, had 60-fold symmetry. And so those were very favorable objects. But a much harder thing, which was also done early on, and that was in both Joachim and Mern, Frank and Van Heel both worked on this, is ribosomes, because they're reasonably big and they have a lot of RNA, so they scatter well, and they're still used for things like that. So this was showing that even though you could barely see anything on the original noisy projections, once you got averages, once you classified them, aligned and classified them into class average, you could see stuff. There were various methods for finding their angles. Angular reconstitution was one that is tricky to use, but it sometimes worked very well. And you could get to 3D, and then you had to reproject and check that the reprojections matched the class averages. And that was, if they didn't, it was wrong. If they did, it might be right. Anyway, so that's where all those ingredients were there. So really all the basic ingredients were there long before all this revolution in technology, which it's easier, I call it the Big Bang. It's easier than revolution. But it was all there. And it was clear that this was going places. And then there was all this improvement. So it wasn't just the direct detectors. It was, you know, the microscopes got much better. There were FEG sources, more coherent. The cryo stages got much more stable. And then there's all the changes to data collection that makes it much faster. There was Chris Russo and others invented nice sample supports that made things move less. And then the direct detectors was this huge leap forward where it was, you could do these movies because they're rapid readout. They were so much faster in the readout. You could take movies and they had tremendous sensitivity and resolution. But the main thing was, until then, we'd either had to use photographic film, which is a real pain and horrendous, or CCD detectors, which were more convenient, but they were rubbish. They were really pretty terrible because they were designed to respond to light, not electrons. So you had to convert the light into electrons, and that was a disaster. And that they took their readout was quite slow. So they were really poor. And some people actually continued to use film for higher resolution for quite a long time, even after the CCDs were available, because it was hopeless. You know, their resolution was terrible. But anyway, all those problems were solved by direct electron detectors. And then people could start getting huge data sets, and the processing really advanced, and Shores and others came along with this using maximum likelihood and nice and machine learning and, you know, all these interesting things. And that is obviously, that's still going on and improving all the time, which is very exciting. And then obviously the map validation interpretation is also going on and improving, and all this is now and being automated. And I should say, in terms of automation, Bridget Carragher was one of the pioneers in that area. And Dan, Claire and I went to Scripps at the time to use Legend on, because we wanted a big data set. I mean, it was on CCD. We had to collect tons of data to get anything decent, but it was the only way to get really a lot of data was to do that by automation. And that was when I was sleeping on the famous sofa, because you had to fill up the side entry holder every 2 hours. And that's when Dave Lander came in as a student, thought I was a tramp that had wandered in off the street and tried to get me arrested, which he didn't even tell me for 10 years. I was so annoyed. Such a good story. Anyway, he eventually admitted it. So that's what put everything in place for where we are now. And this is probably familiar to everybody, that you know, the single particle workflow is really pretty well established, and this is largely automatable. You know, that as long as you have the right samples. So I think the first thing, so there's one step that, well, there are one or two steps where you can't really automate it. You can't automate thinking. You can't, in terms of what should I work on? What's an interesting project? What's worth doing? And then, okay, I want to work on that. How do I make a decent sample that's going to work? That's not going to be falling apart or sticking together or all the things that samples do that you don't want them to do. So that part is still there. You still have to come up with a project that isn't just something somebody else has already done, because it's always easier to do something when you know the answer. If you want to do something new, that's hard. That's the big step. And getting a good sample to do it with is the next big step. Once you've got that, once you've got good grids, then you can really, you know, and sometimes you just fly through all this stuff and get really nice results. So, but the primary steps are still there. And then there's been all these other really nice things. I've been particularly interested in all this classification stuff, and we've already heard about CryoDragon today. I just wanted to, I had this as part of my slides. So if you have heterogeneous things, with continuous variation and all the really tricky problems, which we often have, that, you know, the neural network approaches are really interesting and give very, very nice results. So that's all pretty exciting, I think. And then the tomography workflow, this is trickier because, you know, tilt series are harder. I mean, obviously it's nice that you can do it on a detector and on a film, but, you know, there's a lot of radiation damage is accumulated. And so we have these, you know, dosimetric data collection, so on, that's makes it very effective. And then you have this good data untilted, and you use the high tilt, the lousy high tilt data to figure out more or less where things are in 3D, so that you can use the best bits to do averaging if you can do subtomogram averaging. And that's not so easy because you need tons of objects in the field of view, and that's, you know, your biological object of interest might not be in tons of copies, so that makes it hard. And then this is just another diagram of the idea that you have lots of spikes, or I think these were secretion systems on the surface of a bacterium, and you find them in different orientations, you align them, and you can average them and get a nice subtomogram average. So you've all seen that. One of the tricky things in that is that you can't just treat them like single part. They're nice little 3D particles, but they're not something you can just do cross correlations with, the comparisons between just like that, because they all have a different missing wedge. And you can only, if you're comparing these two here, you can only compare the parts that both have data, the common zone of data. And sometimes that's going to be a very, very small sliver if they're really in different orientations. The missing wedges are going to, you know, take away most of the comparison. So that makes it quite tricky. So all the software has to take care of all this. And that's, yeah, that's not trivial to do, but it can work very well. Obviously the ribosome example with, and the high resolution single particle and the ribosome example of subtomogram averaging were, you know, relatively recent, a few years ago now, very beautiful examples of how this all should work. So, you know, things are obviously at a very nice stage now, and there's lots of interesting things to do, especially on the tomography side, but it's all, you know, pretty nice, I think. So what I thought I'd do for the rest of the time is just illustrate those changes with the work we did, because we started working on GrowEL quite a long time ago, and then I'd left it. This was all long before this resolution business, the Big Bang. So I started very early on, and then we kind of moved on to other things, other chaperones and other things after that. And then Scott Gardner, who's a student with me some years ago, came along and said, why not look at GrowEL again now that we've got all the good detectors? And I said, oh, for goodness sakes, I don't go back to that. But anyway, he did persuade me, and he got some, reasonably interesting results. So I thought I would just finish off by showing you how that looked. Again, especially for the younger people in the field, you can't imagine how awful it was at the beginning. So this was the nuttiest experiment ever. So this was, this is negative stain. It was the first, we started looking at GrowEL in the early 90s. And we knew that it was to do with folding. There was something to do with protein folding, but nobody quite knew what it did at the beginning. And, they were trying to crystallize it. And so far, for this point, the crystals hadn't diffracted. And Tony Crowther suggested, we knew it had sevenfold symmetry because you could see these end views with sevenfold, and there were these side views with 4 stripes. And Tony Crowther said, why not take a tilt series just over a 1/7 of a turn, and then find the particles whose sevenfold axes are lined along the tilt axis. Like, we had no idea about how to classify orientations. It was nuts. So we did that on film, photographic film. And Alan Roseman did the processing. And there were very few particles that were aligned the right way. This was hard work. And then one of them gave the best reconstruction. So this was the reconstruction. And I went and showed that at the Gordon conference that year, and people thought I was absolutely nuts because it was one molecule that we'd done this tilt series of times 7, 6 views times 7 or something. So it was bananas. But actually, it wasn't really wrong. It showed us that the molecule had a cage-like structure, and there were things inside, there were spaces inside, because we had no idea. So that was pretty exciting, because the original models, we thought subunits were C-shaped, but they were sticking outwards. So they were like back to back, but it's not true. They're cavity to cavity, and that creates spaces inside these 2 rings of 7 subunits. So that was the first time we saw that. So I thought, people may think I'm nuts, but this is interesting. And then Xiao Chen was in the lab, and she got the cryo tour. While I was at that Gordon conference, we've been trying on an old GL microscope, 120, to get cryo to work. And we didn't know how to get the ice thin enough. But while I was away, I think, she can say she's here too, she got this image. I think this is 80 kilovolts, and you could see the molecules in ice. And that was really exciting, because then you could do cryo single particle. And again, Alan did the processing there. And we still hadn't got to the idea that, you could actually classify to get the different orientations. So these were the different projection views. And then Alan we just back projected it 7 times to get these sort of like stack of 4 bagels type of thing. So we really didn't have much angular resolution, but it was enough to see, you could see what the structure was. And you could see if you cut sections through it, you could see there were spaces inside. And then when GrowES went on top, it was a great big dome. So, we really could see quite a bit of it. And we had, this is at this point, Neil and his colleagues, Tony Clark and his group, Neil was his student at the time, showed up with complexes where they had denatured malate dehydrogenase stuck inside this thing. So malate dehydrogenase is a substrate of GrowEL, and GrowES, and ATP will fold. You can denature malate dehydrogenase, throw it at GrowEL, and then add GrowES and ATP, and you can get folded MDH. So this is, that was pretty exciting. And we actually got density. We had difference maps where we could see the MDH sitting in one of the outer layer of one of these cavities. So that was pretty exciting. So that was 94. I think that was like a September 94. We published this. October 94, Arnowitz and Paul Siegler got the first crystal structure of GrowEL, and we could see then that those cavities were lined by hydrophobic residues. So it really fit together really nicely. So that was quite exciting. So, people started to think, well, maybe cryo has some use, It's a little dubious still. But, and then Alan went on to get, he eventually worked out that if you just use these classification things, which I thought were so boring, you could classify the different side views. And once you had a bunch of side views around the 7-fold, you could get, a reasonably decent 25 angstrom map. And so he got maps. We had different nucleotide states I'm not sure about this ADP one. We never actually got back to that, but ATP did something. They moved around. And when you added the GROES in the nucleotide, then you got this huge change with this great big chamber forming. And then in 97, this was 86, and then in 97, Paul Siegler and Horwich got the crystal structure of the GROEL-GROES complex. And there you can see this is sliced open, and you could see the open cavity has the yellow hydrophobic lining, and the closed cavity is all hydrophilic. So it plays a trick on the substrate protein. It catches it on this nice hydrophobic surface inside the ring of the cavity. And then when you add GROES and ATP, there's a huge conformational change, and the substrate gets jammed inside a hydrophilic chamber, and it has no choice. It can either do nothing or fold, because there's nothing to stick to, and it can't unfold more because it's in a very confined space. It's about 70 angstroms high, that chamber. So that was a pretty good idea of how this thing worked. So, well, what's ATP doing? ATP was really the key to the conformational change, and we want to understand this. So this was the experimented scripts where Dan and I collected this big data set. So these were 60,000 side views, but there were probably, I don't know, must have been 10 times more data, because the side views were quite rare with ATP. It was very hard to get them because, there was a terrible preferred orientation. But anyway, this all got classified into 7 structures. There's all these conformational changes of ATP, and you could put them into a movie. And is this going to play? Yes. So this is looking at one ring. The red and the orange are the hydrophobic lining. And you can see the way the binding sites open up and spread apart. And then there's this big twist, and that final twisted state is where the GROES comes on. And so the key thing here is that the substrate binds to several adjacent sites. So Art Horwich had shown that, substrates that really need GROEL bind to 3 or 4 adjacent hydrophobic sites. So when those things pull apart, there's going to be a force exerted on the protein. So there's a kind of forced unfolding step, and there is evidence for that from other experiments from spectroscopy. So the full protein probably gets pulled apart if it's misfolded, and then it gets dumped in this hydrophilic cavity where it folds or doesn't nothing. If it's released again, and it has another chance to bind and fold. And it's not ATP hydrolysis that does this. It's just ATP binding. The hydrolysis step is to reset things, open the cage, and for stuff to come out, and the next round starts. So it's actually just the binding of energy of 7 ATPs binding that does this. So, I think it's a reasonably interesting. So, and then the next thing was to look at a substrate inside the cavity. Dan got stuck with me for quite a long time. And this was a big viral capsid protein, and you could really see that it was a very big substrate. We wanted big ones so we could, they'd be stuck inside. And this thing is pear-shaped, and it really did fill the cavity. I mean, the side chains and things aren't shown, but we did see this thing, and it looks like the large domain folds, and then it's got a small domain which can't fold in there. But the viral capsomere is assembled from the folded large domain, which folds inside there, and then the small domain folds probably when it binds to its little neighbor. So that all fit together quite well. So that was the end of the story. That and the ATP thing, I thought, right, fine. And then this resolution thing happened, and I was doing other stuff. So and then Scott came along and said, "Oh, come on, let's have another look." And so he did actually, we did look at it again in the direct detector era. It wasn't that phenomenal in resolution because it's still floppy. Though the domains at the outside are floppy, they're wobbly, and obviously they move to accommodate different substrates. So it's not the most rigid object, and it even went out of fashion as a test object eventually. But anyway, Scott we decided to look at rubisco. So bacterial rubisco is a well-characterized substrate of GROEL. It's a classic substrate where there was a lot of biochemical information. So let's see if we can see more about the substrate. And of course, we didn't. The substrate is denatured. You start the experiment by denaturing the substrate. So it's a mess. You're looking at denatured protein, and it's probably a whole mixture of conformations. And I don't know how much data you'd need to sort out individual conformations of that, but it's just really messy. So we didn't get high resolution. We didn't get interpretable structure on the rubisco, but we did get a lot of detail on what it's binding to on the GROEL, and that wasn't always the residues we expected. So that was quite interesting. But what I think probably the most interesting thing he found, well, 2 interesting things, was he managed to trap a nucleotide-bound intermediate with no GROES. And this is the one where it really was interesting. So there's a eukaryotic version of this called CCT, which has, it's an 8-fold one. It has 8 different gene products, and it works in an asymmetric way to fold actin and tubulin and other stuff. This one is it's a HOMO 14, right? It's 2 symmetric rings of 7. But in this intermediate nucleotide state with rubisco, it's very, very asymmetric. And the ring that binds the substrate, this great green blob is the rubisco, it turned out that about 3 or 4 of the subunits were still binding the rubisco. They were still, the hydrophobic part was still facing the rubisco, but the other 2 or 3 were actually elevated completely in an orientation where the hydrophobic patch was in a GRO-ES acceptor, a potentially GRO-ES acceptor state. So the GRO-ES has mobile loops which dock down on top of those sites. And so it takes away those sites from the bound substrate. And so this was a state where the GRO-ES could land while the substrate was being held in, because we never knew why it just didn't escape, because you had to detach the substrate from its binding sites and put the cap on, and we didn't really know how, what stopped it escaping. So it looks like it really is a hybrid asymmetric structure which traps keeps holding the substrate while the GRO-ES is landing. So that gave us a new mechanistic idea that you have this. Well, there should be 3 of these. Yes, this was an earlier model. So there should be 3 in a possible GRO-ES acceptor state, and then other ones hanging on. So the idea is it's a kind of landing complex for the GRO-ES while the substrate's there. And then he also got a few classes of the encapsulated rubisco. And this was quite nice because this is not the native structure rubisco. It's got 2 domains, and the 2 domains, the folded state at this resolution, the folded state of the 2 domains fits in, but the interface is distorted. And actually, one thing we heard about this morning, I think Wall was talking about hydration water around a protein. Well, we're nowhere near that resolution, but what's very clear is that there isn't room for water in a lot of this interface. This protein the surface of the protein has to get substantially dehydrated to fit into something this tight. So the GRO-EL and the substrate are really jammed up against each other. It really is, it must be very dehydrated on a lot of this surface. If you tried to put in layers of water, you just couldn't there. And I think that's part of the idea. The energetics of this protein folding, of this hydrophobic to hydrophilic change, must also be to do with the water on the surface of the protein. So I think that was quite an interesting thing. And then this interface, as I said, it wouldn't fit in. The native interface would make the little domain too far down. So it seems to be distorted to fit in there. And then when it's released, it probably readjusts the interface. And then this is the final, what these domains look like when the GRO-ES is actually landed on them. So that's the story. I think I'm just about done. So just to summarize where we are with this field, obviously there's been tremendous progress, but there's plenty of challenging things still around. I think preferred orientation is still a plague. I should say Scott used the chameleon to get those GRO-EL ATP structures, which helped. Didn't make it perfect, but it certainly helped because it was very hard. The ATP bound ring just splats down onto the interface and it gets deformed. It just gets wiped out. It really is bad. Neil struggled for a long time with that. So that's still a problem. There's no perfect fix to that. Obviously, there's lots of wobbly, heterogeneous stuff. And the most interesting things are the most wobbly and heterogeneous. Those are the things that are doing things. So that's where a lot of effort is going in, and it should go in. There's a lot of, you need a lot of computers. One thing that we have worried about over the years is, especially when we're doing this classification where we're really only coming out with a tiny subset, is this biochemically valid? We're taking a tiny minority of the sample to make our structures. I think with these new methods of looking at the continuous heterogeneity and so on, that probably helps with that, but we certainly do that quite a lot. I think 3D classification is still not that straightforward. So the 2D classification, you can make this graph, from the original MSA, but 3D is more complicated because there's projections and the classification to sort out the conformational variations. So these things are still not that straightforward. Model building seems to be getting very good if you have decent resolution, which often we don't. And then, in terms of biological interpretation, let's go back to the thinking part. So there's been a lot of focus on what's the resolution, and it depends what it tells you. So I'm all in favor of saying, well, what does it show rather than what's the FSC value? Yeah. So I think the real things that gets back to our experimental design, formulating interesting questions to ask and how to answer them. And there's obviously all the stuff with tomography. And one of the things I found quite frustrating about tomography of cells is, you know, tomograms give the answer, but you don't know what the question was. You don't know what it is you're looking at. Now, obviously, this morning we heard of a very fantastic story of how you could find out from the tomogram what the stuff was, but that's really pretty hard. I was very impressed that could be done in those cases. And then there's all this machine learning stuff, which is going to solve all our problems. So, so I think I'll just wind up there. So over the years, so there was a visitor from China early on who collected that first partial tomogram on film, photographic film. And then Shasha and Alan came along and did a lot of what I talked about, and then Neil and Dan, and then Scott. So, and we've had a lot of support over the years from Natasha and Dave Holdeshaw in our lab. And that original work with Neil was with Tony Clark from Bristol when we started looking at substrates. And then we did a lot more work with substrates in different states with Art Horwich and mutants. And then that hilarious expedition to Scripps was Percy Bridget Carrier. So thanks for your attention. And I don't know if people want to discuss this, where it should go next, or maybe you're certainly going to have to discuss it tomorrow if we don't discuss it now.